So what is granuloma cells? Granuloma cells are specialized immune cells consist of mature mononuclear phagocytes that then form a structured compact collection. Mature mononuclear phagocytes consists of macrophages, epithelioid cells and multinucleated giant cells. The function of granuloma cells is act as localized immune response that isolate and contain persistent pathogens, foreign substances or irritants that are resistant to normal immune response. It also helps in maintaining tissue integrity. As per mentioned before, granuloma cells consist of epithelioid cells, macrophages and multinucleated giant cells. So where does these cells actually come from? And what is the origin of granuloma cells? All those cells begin from blood monocytes, which will then differentiate to tissue monocytes or immature macrophages when leaving the bloodstream. After being triggered or activated by chronic infection or foreign materials that cannot be effectively cleared, those immature monocytes will then further differentiate to phagocytic macrophages or mature macrophages and epithelioid cells. Then, the epithelioid cells will sometimes fuse and form multinucleated giant cells. What are epithelioid cells? Let's begin by understanding epithelioid cells. Epithelioid cells are specialized macrophages that resemble epithelial cells in appearance. They are also referred to as epithelioid histiocytes. These cells are derived from monocytes, which differentiate into macrophages. Under chronic immune activation, such as persistent infection or inflammation, these macrophages further differentiate into epithelioid cells. The morphological characteristics of epithelioid cells are distinct in their structure. They are large, elongated cells with abundant pale pink-stained cytoplasm under the microscope. Their nuclei are oval or elongated. Unlike macrophages, they tend to form cohesive clusters resembling epithelial sheets. This arrangement contributes to the structural organization of granulomas. Their primary role is secretion rather than phagocytosis. They produce cytokines and enzymes that regulate inflammation and attract other immune cells. Epithelioid cells are a hallmark of granulomas, which are small, organized collections of immune cells. In granulomas, epithelioid cells cluster together in the center. They may also fuse to form multinucleated giant cells, which are another key component of granulomas. Together, these cells wall off pathogens or irritants that the immune system cannot destroy, effectively isolating them. So when do epithelioid cells appear? Typically form in response to chronic inflammation. They are seen in granulomas associated with: first is infectious diseases, like tuberculosis, leprosy, fungal infections, and brucellosis. Second is autoimmune and inflammatory disorders like sarcoidosis, a granulomatous disorder of unknown cause, and Crohn's disease, a type of inflammatory bowel disease. Third is foreign body reactions. These occur when the body reacts to persistent foreign materials like sutures or implants. Lastly is other granulomatous disorders like granulomatosis with polyangiitis and berylliosis. Epithelioid cells are critical in granuloma formation as they help wall off pathogens or irritants when they cannot be effectively cleared by the immune system. Next is multinucleated giant cells. Multinucleated giant cells, or MGC, are large cells that form through the fusion of multiple macrophages. They are commonly found in response to chronic inflammation or infection. These cells play a significant role in the immune system by engulfing pathogens and remodeling tissue. They contribute to granulomas, which are structures that form during chronic infections or inflammations. The formation of multinucleated giant cells starts with inflammation. They form during chronic inflammation. When the body detects an infection or a foreign object, it triggers an inflammatory response that brings in monocytes. Second is macrophage activation. The monocytes move to the site of inflammation and turn into macrophages. These activated macrophages help fight infections by attacking pathogens and removing debris. Lastly is macrophage fusion. When the inflammation continues, these activated macrophages fuse together to form multinucleated giant cells. This fusion is driven by certain signals include cytokines and adhesion molecules. Based on their morphology and functional characteristics, there are generally 4 types of multinucleated giant cells including osteoclast, foreign body giant cells, Langerhans giant cells, and Tooten giant cells. Conditions associated with multinucleated giant cells include bone-related diseases like giant cell tumor. Second is granulomatous infections like sarcoidosis. Lastly is infectious diseases like measles. Next is the differences between epithelioid cells and multinucleated giant cells based on their characteristics. First, of characteristics for epithelioid cells. The nucleus of the cells appears to oval or elongated with fine to coarse chromatin and prominent or inconspicuous nuclei. while the cytoplasm appear to have round or spindle-shaped with abundant or scanty cytoplasm and foamy appearance. So the appearance of epithelioid cells are not fixed. Their shape changes with activity. In a resting state, they are more rounded with abundant cytoplasm, but when activated, they become elongated or spindled with scanty cytoplasm. Another is the presence and absence of foamy appearance, which also depends on the activity of the cell. The cell will appear more foamy when they are actively phagocytosing. Second is the characteristics for multinucleated giant cells. So, for MGCs, the cells appear to have multiple nuclei. Pattern arrangement of the nuclei based on the type. The first type is Langerhans giant cells, where it has a horseshoe or ring-like arrangement of their nuclei. They commonly associated with TB or sarcoidosis. The second type is Teuton giant cells, where it has ring of nuclei surrounding lipid halo area at the peripheral of the cell. They commonly associated with xanthomas. The third type is foreign giant body type, where it has nuclei scattered throughout the cell. They commonly associated with implants or foreign body reactions. The fourth type is osteoclast giant body type, which are found in bone-related conditions and tumor-like giant cell tumor of bone. Characteristics of the MGC cytoplasm is abundant, and also it is larger than most cells with foamy appearance. Now is the differentiation of epithelioid cells and multinucleated giant cells based on its mitochondrial. Let's begin with what is morphometric analysis. Morphometric analysis is quantitative analysis, useful for an objective assessment of cellular variations. In terms of cytology, it is a quantitative method that evaluates measurable cellular and nuclear features to detect pathological changes, assess disease progression, and monitor the effectiveness of treatment. Now let's see the differences of epithelioid cell and multinucleated giant cell based on the mitochondrial. It only has minor differences in the mitochondrial profile section. Based on the mitochondrial shape, the epithelioid cell is spherical rather than a multinucleated giant cell. Multinucleated giant cells are elongated. It is related to the second point, which is numerical eccentricity of mitochondrial section profiles. Numerical eccentricity of mitochondrial section profiles refers to the shape of mitochondrial section profiles, quantifying how stretched or elongated they appear. For the epithelioid cell, the numerical eccentricity of mitochondrial section profiles is 0.658 per 0.16 mean per standard error of mean, which means that less stretched makes the shape become spherical. For multinucleated giant cells, the numerical eccentricity of mitochondrial section profiles is 0.723 per 0.06 mean per standard error of mean, meaning it is more stretched, that why the shape is more elongate. This component is important because the shape of mitochondria can relate to their function. More elongated mitochondria may indicate increased energy production, but for rounder mitochondria could suggest a lower energy state. Last one, volume density of mitochondria. Epithelioid cells have 0.052 per 0.004 mean per standard error of mean, meaning it has low volume density. However, for a multinucleated giant cell, it has a high volume density of 0.059 per 0.005 mean per standard error of mean. When volume density of mitochondria increases, it shows cells need more energy, so it will produce more mitochondria to meet the energy demand. So, by elongation, the mitochondria increase their surface areas in relation to their volumes. It is possible that the alterations in shape are associated with enhanced activity of membrane-bound enzymes and an increased exchange of substances between the mitochondria and cytoplasm. Next, we will look into the differences between epithelioid cells and multinucleated giant cells based on their overall lysosomal size. According to the study, there are 3 parameters used to disclose the differences, which are the lysosomal numerical density, that refers to the amount of lysosomes in 1,000 cubic micrometer of cytoplasm, diameter range, and volume density, which refers to the amount of space occupied by the lysosomes. Based on the result, we could conclude that blood monocytes have moderate amounts of lysosome with small size, tissue monocytes have less amount of lysosome and in moderate size. Then, epithelioid cells have abundant lysosome and they are in moderate size, whereas phagocytic macrophages have moderate amount of lysosome that are in large size, and lastly, multinucleated giant cells have abundant amount in moderate size of lysosomes. Next, for better visualization of the data, the study also presented a graphical representation of the cumulative sum of numerical density of lysosomes, whereby epithelioid cells being the cell type that contain the highest number of lysosomes, followed by multinucleated giant cells, blood monocytes, tissue monocytes, and finally phagocytizing monocytes. Therefore, several findings were concluded in the study. Epithelioid cells and MGC have high lysosomal numerical density, may indicate their increased synthetic and digestive performances. Also, MGC contain more small lysosomes than of epithelioid cells, which suggest a continued and enhanced metabolic activity following cell fusion. Both cells may also influence the immune cellular microenvironment of the granuloma, and finally, active usage of lysosome during phagocytosis may contribute to the large lysosomal size in phagocytic macrophages. To conclude, granuloma cells are a structured immune response that is a hallmark of chronic inflammatory diseases such as tuberculosis and sarcoidosis. With the help of morphometric analysis, epithelioid cells and multinucleated giant cells can be differentiated by evaluating the measurable cellular and nuclear features of cells. Granuloma formation is an essential response to persistent stimuli, while morphometrics analysis is a crucial tool to understand the development, progression, and potential treatment for those cells in response.